Good morning, everyone. Today, we're diving into a fascinating journey, exploring what we call the ascorbate paradox. This isn't just about a single molecule, it's about metabolic plasticity that has unfolded across eons, fundamentally shaping the evolution of plants, primates, and even parasites. Ascorbate, or vitamin C, is an essential cofactor, a critical antioxidant and enzyme helper required by almost all eukaryotes. Its biosynthetic history is a remarkable tale of adaptation to oxidative stress and the acquisition of new cellular components. The evolutionary problem it addresses is complex. The pathway to vitamin C is anything but linear. It has been re-engineered, lost, and even substituted multiple times to solve specific biological challenges, particularly reactive oxygen species toxicity and shifts in dietary habits. We'll trace three distinct strategies: the switch in plants, which replaced a dangerous enzyme; the trade in primates, where synthesis was traded for recycling; and the trap in parasites, revealing a fatal vulnerability due to strict reliance on synthesis. These adaptations define the health and vulnerabilities of every species. Let's start at the ancestral baseline, tracing back to the last common eukaryotic ancestor. Vitamin C synthesis relied on an enzyme called L-gulonolactone oxidase, or GULO, which was localized to the endoplasmic reticulum. However, this ancestral mechanism had a significant flaw. GULO uses molecular oxygen as an electron acceptor, producing hydrogen peroxide as a mandatory waste product. Hydrogen peroxide, as many of you know, is a potent reactive oxygen species. This GULO-dependent synthesis was functional in basal metazoans and fungi, as you can see marked on our phylogenetic tree. But it was lost in insects and, notably for us, in higher primates. The original machine, GULO, literally converted L-gulonolactone with oxygen into ascorbate, but always with that toxic byproduct, H2O2, tagging along. This brings us to a major turning point: the chloroplast conflict. The acquisition of the plastid, or chloroplast, was revolutionary. It enabled photosynthesis, transforming life on Earth. However, this process also generated massive amounts of reactive oxygen species, or ROS. The incompatibility was striking. Using the ancestral GULO enzyme to produce the antioxidant vitamin C actually created more ROS in the form of hydrogen peroxide. This made the GULO pathway metabolically dangerous for any photosynthetic organism. Imagine a plant cell constantly battling this internal overload, where the very mechanism meant to help combat stress was actually contributing to it. There's a curious exception: cyanophora, a type of glaucophyte, retains GULO but doesn't use ascorbate for photoprotection, relying on alternative mechanisms instead. So, how did photosynthetic organisms resolve this conflict? Through what we call the great enzyme switch. The ancestral or animal strategy, as we've discussed, involved GULO, which led to ascorbate but also to toxic H2O2. The solution for plants and algae was nothing short of brilliant endosymbiotic gene transfer. Photosynthetic lineages replaced GULO with a new enzyme, L-galactonolactone dehydrogenase, or GLDH. This enzyme was likely transferred from the symbiotic organism, which became the chloroplast, to the host nucleus. The distinct advantage of GLDH is its mechanism. It uses cytochrome C as an electron acceptor. This completely uncouples ascorbate synthesis from the production of reactive oxygen species, making the process safe and efficient, without the toxic byproduct. While the GLDH enzyme became universal, the specific pathway to produce the substrate for GLDH actually diverged. In plants and green algae, the non-inversion pathway is used, starting with D-mannose and moving through L-galactose to ascorbate, utilizing the VTC2 enzyme. However, in euglenids, which acquired their chloroplasts through secondary endosymbiosis, a different, inversion, pathway is seen. Here, D-glucose leads to D-galacturonate, which then becomes ascorbate. This is a hybrid pathway where the C1 atom becomes C6. Despite these differences in chemical routes, the universal convergence on the safe GLDH enzyme, localized in the mitochondria, is a striking example of evolutionary efficiency across all photosynthetic eukaryotes. Now, let's turn to a fascinating, and for us, quite relevant, development: the primate deficit. This is a classic example of what we call ascorbate oxytrophy, or the use it or lose it principle in evolution. The primary cause for primates losing the ability to synthesize their own vitamin C was the sheer dietary abundance of GLDH-derived vitamin C from plants. This relaxed the selective pressure on the GULO gene. If you're constantly consuming something essential, the internal machinery to produce it can become redundant. The evidence is compelling in primates. The GULO gene hasn't completely vanished, it remains as a non-functional pseudogene, a genetic fossil. In insects, there's been a complete loss of GULO, and remarkably, bats show multiple independent loss events, highlighting this evolutionary pattern across different species. So, if primates lost the ability to synthesize vitamin C, how do we get it? This leads us to a remarkable compensation mechanism, the GLUT1, inborn error, adaptation. Humans, in essence, switched from manufacturing vitamin C to becoming expert recyclers. To illustrate this, let's compare mouse and human red blood cells. A mouse RBC actively synthesizes about 200 mg per kilogram of vitamin C. In contrast, human RBCs have synthesis blocked. Instead, they upregulate GLUT1, a glucose transporter protein. But here's the clever part: GLUT1 preferentially transports dihydroascorbate, or DHA, which is the oxidized form of vitamin C, over glucose. This turns our blood cells into a circulating antioxidant reservoir, scavenging and recycling DHA. Our dietary intake is comparatively modest, only about 1 mg per kilogram, a testament to our recycling efficiency. This sophisticated recycling engine involves another key player, stomatin, and what we call the DHA switch. Stomatin is a protein that modifies the behavior of GLUT1, specifically enhancing its preference for DHA uptake over glucose. Imagine it as a finely tuned gatekeeper. Once inside the cell, this oxidized DHA is immediately reduced back to its active form, ascorbic acid, ready to function as a powerful antioxidant. The efficiency is astounding. We have clear evidence of stomatin's crucial role. Patients who are stomatin deficient show a significant 50% drop in DHA transport, underscoring its importance in our ability to maintain circulating vitamin C levels. Now, let's discuss another intriguing substitution that occurred in human evolution: the uric acid trade-off. Looking at our evolutionary ledger, humans have made some interesting debit and credit entries. On the debit side, we lost the urate oxidase, or uricase, gene. This gene is responsible for degrading uric acid. Its loss occurred via nonsense mutations. The consequence? On the credit side, we gained significantly higher plasma uric acid concentrations, approximately 10 times higher than other mammals. This wasn't a random change. Uric acid is a potent antioxidant scavenger. What's truly remarkable is the co-evolutionary aspect. The loss of the uricase gene correlates strongly with the earlier loss of the GULO gene. In essence, humans replaced high-volume vitamin C synthesis with high-level uric acid retention as a primary antioxidant defense. So, why was retaining high levels of uric acid so beneficial? It acts as a neuroprotective shield. The chemistry is precise. Uric acid specifically scavenges harmful singlet oxygen and peroxynitrite, protecting our DNA from single-strand breaks. This is especially crucial for the brain, an expensive tissue, with high oxygen use and a rich lipid content, making it particularly vulnerable to oxidative stress. Clinically, we see compelling correlations. Patients with multiple sclerosis and Parkinson's disease consistently show lower serum uric acid levels. Conversely, higher uric acid levels are correlated with a reduced incidence of Parkinson's disease. This suggests uric acid plays a vital role in safeguarding our neurological health. However, like many adaptations, this comes with a double-edged sword. While the survival benefits were clear: significant neuroprotection, enhanced antioxidant capacity, and likely a prolonged lifespan, there were also biological costs. High uric acid can precipitate as crystals, leading to gout, a uniquely human affliction. It's also implicated in hypertension and increased cardiovascular risk. So, why did evolution favor this trade-off? The evolutionary math is clear: the protection against oxidative stress and the potential for an increased lifespan outweighed the risks of conditions like gout during human evolution. It was a strategic decision that shaped our species' health profile. Finally, let's explore the trap strategy, specifically the parasitic vulnerability of Trypanosoma cruzi, the causative agent of Chagas disease, which affects millions, particularly in endemic zones like parts of South America. Like humans, T. cruzi has a sophisticated defense mechanism. It relies on an ascorbate-dependent peroxidase, or TCAPX, to detoxify immune system attacks. The critical problem for this parasite is that it absolutely requires vitamin C to fuel this defense system. This dependency creates a fascinating vulnerability, one that we can potentially exploit. Here's why this is a trap. Our transport assays provide clear evidence, a smoking gun, if you will, that T. cruzi is utterly incapable of scavenging vitamin C from its host. The graph clearly shows that while glucose uptake is robust, ascorbate or DHA uptake is negligible. This leads us to a crucial conclusion: the parasite is metabolically isolated when it comes to vitamin C. It relies entirely on de novo biosynthesis, meaning it has to make its own. If this synthesis pathway is blocked, it has no backup, creating a fatal vulnerability for the parasite. This metabolic isolation makes T. cruzi's biosynthetic pathway an ideal target for chemotherapy. The key enzyme here is TCGAL, or galactonolactone oxidase. This enzyme is remarkably active, more than 20 times more active than its counterpart in related parasites like T. brucei. Its mechanism involves the non-covalent binding of an FMN cofactor. The profound vulnerability is that humans lack this pathway entirely. This presents a unique opportunity. By inhibiting TCGAL, we can effectively starve the parasite of its essential vitamin C, offering a chemotherapy route with minimal host toxicity because the human body doesn't possess this enzyme. In summary, the history of vitamin C illustrates three profound strategies for survival, demonstrating incredible metabolic plasticity. Plants employed the swap, replacing the dangerous GULO with GLDH, resulting in safe, ROS-free synthesis. Humans adopted the substitution, losing GULO but gaining highly efficient GLUT1-mediated recycling and enhanced uric acid as an antioxidant, leading to high-efficiency scavenging. And for parasites like Trypanosoma cruzi, the trap emerged. They retained their own unique synthesis pathway via TCGAL but cannot scavenge vitamin C, creating a critical therapeutic vulnerability. Ultimately, the story of vitamin C is a record of how oxidative stress has continuously reshaped eukaryotic biology. Adaptation isn't just about simple changes, it involves complex gene transfers, strategic trade-offs, and intricate compensations that ultimately define the health, resilience, and vulnerabilities of every species on Earth.